Thanks everybody for coming today. Z Evolution for today is presented by Kaitlin Higgins. Kaitlin is a PhD candidate in my lab and started grad school in 2019. So she's going to be talking about maize mdr1. So take it away, Kaitlin.
Thank you. So I've titled this talk Exploring Maize Endosperm Transcription Regulation through a Mutant of the Glycosylase mdr1. And it's a mouthful, so don't worry, I'm going to give you a lot of background. And so first, I want to give you an overview about what I'll be talking about. The first will be the background, and then I'm going to tell you about how differentially methylated regions overlap differentially expressed genes more frequently than non-differentially expressed genes. Then I'll tell you a little bit about how transposable elements are involved in the regulation by mdr1. And finally, I'll talk about how differentially expressed genes are frequently also imprinted and display maternally biased expression. So because not everybody thinks about the genetics of the seed as frequently as I do, I wanted to give everybody a refresher on what that looks like. So maize undergoes double fertilization, and that's because the pollen actually has two sperm cells. One goes in and fertilizes the egg cell, resulting in a diploid embryo that will grow into the mature plant. And the other sperm cell fertilizes the central cell, which actually already has two copies of the maternal genome. So this results in a triploid endosperm. But before all of that happens, the central cell actually undergoes widespread demethylation. And this leads to a distinct transcriptomic environment in the endosperm, which can also is partially influenced by something called genomic imprinting, or it influences genomic imprinting. And genomic imprinting is expression of a gene dependent on which parent it is inherited from. And so one of the first imprinted genes discovered was R1. And so I have a figure of that right here. And so what we're seeing is a reciprocal cross. On the left side, we have W22 being the maternal donor and B73 being the paternal donor. And on the right side, we have B73 being the maternal donor and W22 being the paternal donor. And as I mentioned, the central cell undergoes widespread demethylation. So what happens is these methylation lollipops that are present on the paternal genome aren't there on the maternal genome, and so that ends up being expressed. And in this cross in particular, we have purple ears when W22 plays the maternal role. And when B73 plays the maternal role, we actually end up with a mottled ear. So we identified these using reciprocal crosses and then harvesting endosperm from each side of the reciprocal cross. And this initially was discovered way back in 1970, but nowadays we're able to actually gather all of the genomics data and then show differential expression of the same genes when they're inherited maternally versus paternally. And maternal derepression of R1 is a regulator of R1, which I just told you about. And this is a mutant it's a mutant in which the characteristic expression of R1 is disrupted. And so what I've highlighted over here is W22 being selfed with the wild type mdr1 and wild type R1. And this results in a purple ear as we would expect. But with the mutant mdr1 in a selfed W22, we end up with a mottled ear. And that's because R1 doesn't have the methylation removed from even the maternal genome. So it only has leaky expression. And recently, working with Jonathan Gent at the University of Georgia, we mapped this mutant and identified differentially methylated regions. And we found that this is homologous to Demeter and Arabidopsis, which has been implicated in imprinted expression. So for the purposes of this talk, I want to define differentially methylated regions. So these are regions that have different levels of DNA methylation in the CG, CHG, and CHH context. And so in this figure, I have methylation lollipops again, and we say that state B is the wild type state of the endosperm. then we see transcription occurring. And then state A might be one version of the mdr1 environment where there's methylation now present on the transcription start site. And so this would be a DMR between wild type and mdr1. And it's in a location that prevents transcription in the mutant. But DMRs can occur other places as well. In this case, between state B and state C, there's a DMR that's really far upstream of the gene and doesn't actually end up affecting transcription of that gene. And so I wanted to just make sure that we're all on the same page about how differentially methylated regions can lead to gene repression if they're covering controlling regions of the gene in methylation. And we looked at differentially methylated regions between wild type endosperm and mutant endosperm. And I mentioned Demeter a couple slides ago, so I just wanted to give you some background on what we know because of that mutant. So even though imprinting was discovered in maize, there's been a lot of work done to study it in Arabidopsis. And so we already have some ideas of what we might see in maize, but it's all pretty exciting. So in Demeter, or Demeter in Arabidopsis is required for seed viability and imprinted expression. And the loss of it reduces demethylation of transposons and companion cells. And they've also found that it's primarily responsible for widespread demethylation of the central cell that I mentioned at the beginning of this talk. So our design was to self wild type W22 and mutant W22, and then we dissected 10 endosperm from 15 days after pollination cobs. And in previous work, we ran methyl-seq and identified differentially methylated regions. And recently we ran RNA-seq and identified differentially expressed features. So throughout this, I have pulled in previously published data from Ghent et al. 2022, which is where I'm getting the methylation data, and Anderson et al. 2021, which is where I get the imprinting data. And so now I'll tell you about the differentially methylated regions and the overlap with differentially expressed genes. So as I mentioned, we previously published with Jonathan Ghent at UGA and identified high levels of differential methylation between wild type endosperm and mdr1 mutant endosperm. And so in this figure, what we're looking at is methylation levels in the three contexts with CG at the top, CHG in the middle, and CHH at the bottom. And the blue line is the methylation level in wild type endosperm, and the gray line is wild type embryo. And so we see that in all three contexts, that blue line is a lot lower than the gray line. And this would be the widespread demethylation that we see in the central cell, causing this difference. But if we look at the orange line, which is the mdr1 endosperm, in the CG context, it is much closer to the embryo levels of methylation. And in the CHG context, it's also nowhere near as demethylated as the wild type endosperm. And so this is just showing that mdr1 is responsible for a lot of demethylation in the central cell. And so when we started looking at the differential expression data, we unsurprisingly found that the majority of differentially expressed features were downregulated. We had 96 genes downregulated and 86 transposons downregulated. And that I say unsurprisingly because what we kind of expected was that with the methylation still being in place, a lot of genes wouldn't be able to be transcribed. So one of the first things I asked was, where are these differentially methylated regions we've already identified in comparison to these differentially expressed features? And what we found was that DE features tended to have overlapping DMRs at higher rates than non-DE features. And in this figure, this is just the frequency of the occurrence of where a DMR is. So we have different classes. In green, we have overlapping. So the DMR is within the annotated gene or TE. And then in yellow, we have more than 2 kb away, and in pink and red are within 2 kb of the gene. And so just looking at the green and the yellow, we can see that differentially expressed genes have overlapping DMRs much more frequently than non-differentially expressed genes. And non-differentially expressed genes tend to have DMRs that are a lot further away. And it's similar with TEs. So thinking of TEs, we had a lot that were differentially expressed. So we wanted to look at that a little bit more, especially knowing the involvement of Demeter with demethylating transposable elements. And since this is another thing not everybody thinks about all the time, I wanted to just do a little bit of a refresher for the different types of transposable elements. So we have 2 major classes of transposable elements. We have DNA transposons, which are also thought of as the cut and paste. And we have the RNA transposons, which are thought of as the copy and paste. And of these, they can also be subdivided into different orders. And these are the common names people usually know. So DT is an abbreviation for terminal inverted repeats. We also have helitrons and long terminal repeats, and then interspersed nuclear elements. However, in maize, there's only 3 of those orders are really important. And those are terminal inverted repeats, helitrons, and long terminal repeats. And those can further be subdivided into superfamilies and families and then copies. But it's important to understand how much genome space is taken up by each of those 3 groups. So in purple over here, this is the amount of genome space covered or taken up by long terminal repeats. And then in green, we have terminal inverted repeats, and in blue, we have helitrons. And so one of the first things I did when we were looking at the TEs was to ask which superfamilies they belong to. And so out of the 86 TEs that are downregulated, we only found 4 superfamilies that had more than 5 members called as differentially expressed. And each of these dots is a different replicate. And so there's 6 replicates in each column, and each of these columns is a different downregulated TE. And so we see here that we have a lot of helitrons, or the DHH superfamily. And the orange-brown color is the wild type expression levels of each of those transposons. And the red is the mutant levels of expression. So we see that expression drops to pretty much 0 in the mutant. And the other families that we find with more than 5 members called as differentially expressed are all long terminal repeats. But we still see a similar pattern with most of the expression levels dropping to nearly 0 in the mutant. So I went in and got a little bit more specific with what we were looking at with the TEs, and found that there are only 2 families that have more than 5 members called DE. So last slide, I was looking at the superfamilies, which can contain a lot of different types, but this is just families, which are, a further subdivision. And so this is similar to the last plot with looking at expression, except now there's also shapes. And so the 2 families we found were DHH2, which is a helitron family, and that's the pink circles and dark triangles. And as we can see, that's a lot of the ones that are called DE. And the other family was a long terminal repeat family. And so I wondered in general, how many differentially expressed genes have overlapping TEs that maybe weren't identified as the TE when we were mapping the reads. And so I asked, how close are the closest TEs to all of these DE and not DE genes? And we found that DE genes more frequently have an overlapping TE than non-DE genes. So my next question was, well, what types of TEs are overlapping these genes. And so I plotted the closest type, the superfamily of TEs to the differentially expressed and non-differentially expressed genes. And there's a lot of differences here, but one thing I wanted to highlight is that there's a big increase in the number of helitron families, or helitrons that are the closest type of TE to the differentially expressed genes. And that out of 127 TEs that were close to the 96 DE genes, 40 of those were helitron superfamily, and 31 of those 40 were DHH2 family transposons, which was also, the largest group of family that, or the largest family that had a bunch of DE transposons in this study. So what's so special about DHH2 family transposons? Well, first, in a recent paper from Michelle Stitzer, the authors predicted that the DHH2 family transposons are the only family to have autonomous members in B73. And in our previous paper, Anderson et al. 2021, we showed that 44 members of the DHH2 family have maternally or are maternally expressed. And then in another paper from Batista et al., they suggest that helitrons are responsible for imprinted expression through the movement of a specific class of binding sites in Arabidopsis. So the same could be happening in maize. And now that I've brought up imprinting, we're going to finish out this talk by discussing that a little bit. So as I mentioned at the beginning of the talk, genomic imprinting is expression of an allele dependent on which parent it is inherited from. And in 2021, we published a paper identifying hundreds of genomically imprinted genes across 3 maize lines, including W22, as well as looking at imprinted transposons. And so this figure is from that 2021 paper and shows how many genes were called as maternally expressed genes or paternally expressed genes in each of these crosses. So W22 would be the present in BW, but the first line here is when it's playing the maternal role. So we have 117 that are maternally expressed when crossed with the B73, and 108 that are maternally expressed when crossed with PH207. But then when we look at the transposable elements, we see a very low rate of paternally expressed TEs, but a lot of maternally expressed TEs for all 6 crosses. And that's important because we end up seeing a lot of maternally expressed features with this study. And so one of the first things that I wondered was how many of these are both imprinted and differentially expressed. And so this is just a quick Venn diagram of what we found. And so out of all of the MEGs and DE features, there was an overlap of about 40% of the DE features being imprinted as well for both genes and TEs. But then I started asking questions like, are there differences in the level of expression between DE genes that are also imprinted and those that aren't imprinted? And I wondered if cutoffs for imprinting are inhibiting our ability to see overlaps. So to answer the first question, I plotted the log2 RPM, or reads per million, of the DE features in wild type and the mutant, and then I separated them based off of whether they are imprinted or not imprinted. And what we found was that in wild type, the imprinted features, the only ones we found were maternally expressed. So that's why there's no paternally expressed. But these imprinted features are significantly, or expressed significantly higher than non-imprinted features. And in the mutant, they dropped really low. So there's not very much information there. But yeah, so the answer to that first question is that they are expressed at higher levels than non-DE features. And the next question about whether our cutoffs were inhibiting our ability to see this is a question that I had because we use very strict cutoffs to determine imprinting. And so I wondered if that was influencing our ability to see the full influence of mdr1 on imprinted expression. And so what I did was I pulled in data from the 2021 paper about the log twofold change between when a gene's inherited maternally versus paternally. And I compared that to our DE and non-DE genes. So this, what I pulled in is essentially a proxy for imprinting from that paper. And so DE is in this red-brown color and yellow is not DE. And typically because endosperm is triploid and has 2 copies of the maternal genome, we would expect a log twofold change for biparentally expressed genes to sit at around 1, which is this where this red line is. And that is what we see is generally true for non-DE transposable elements and genes. But when we look at the differentially expressed features, we see this strong push towards a positive log twofold change. And because of the way these tests were set up, maternally biased expression is positive values of a log twofold change, and paternally biased expression is negative values. And so what this means, especially with having such a significant difference between differentially expressed features and these log twofold change and non-DE, is that mdr1 is influencing a maternally biased expression pattern, which is really exciting. And because I gave you so much information, I wanted to kind of summarize it using some alluvial plots. So what I have showing here is a non-differentially expressed gene, or all the non-differentially expressed genes. And I have 3 levels that it's been sorted into, and each of these little lines is going to be 1 gene. So the top row is how far away the closest differentially methylated region is to that gene. The middle row is the genomic imprinting status. So we have very small lines. That's all. The paternally expressed gene 1 is almost imperceptible, as is the maternally expressed gene 1. And finally, at the bottom, we have whether they are synthetic with sorghum or not. And so if we look at the differentially expressed genes, we see a big shift in all 3 of those lines. In the closest DMR, we see a big jump in the closest DMR being overlapping, a big jump in the number of maternally expressed genes, and a big jump in the number of these features that, or these genes that are not synthetic with sorghum. And it's pretty similar for transposable elements, which we did not assess for synteny with sorghum because there's so much presence-absence variation. And so in conclusion, the main points of this talk are that mdr1 is homologous to a well-studied protein in Arabidopsis, and we recently mapped it and used a mutant to delve deeper into understanding its function. And through that, we discovered that differentially methylated regions overlap differentially expressed genes more frequently than non-differentially expressed genes, that transposable elements are involved in the regulation by mdr1, and DE genes are frequently also imprinted and display maternally biased expression. And so with that, I just want to thank Jonathan Gent and his team at the University of Georgia, my PI, Sarah Anderson, and our lab tech, Connor Valentine. Thank you.
Nice work, Kaitlin.
Thanks.
What questions do you guys have? I see Ed has put a question in chat. Do you want to ask your question, Ed?
Yeah, I was just kind of wondering for the differentially expressed genes, how many, do you have an estimate of what proportion of them actually show up in the proteome?
I don't think we do. I haven't looked at that.
Yeah, this is one of these questions we keep on asking for all these genes out there. You're kind of like going back to that Wally paper and just saying, okay, how many of them actually show? Do we have? I mean, obviously, there's going to be a whole bunch of them just because of sensitivity don't show up in the proteome, right? But is that proportion of proteome really different between the ones that show up as where you think you've been able to score as differentially expressed versus non-differentially expressed? And we just keep on wondering how much translation is the blocking point, and some of this is noise, some of it isn't noise, and the whole process.
Yeah, I mean, I think that's probably a good idea to do, because we've been using synteny for like real genes to determine which ones are important. And yeah, it could be interesting to look at that since so many of the differentially expressed genes are not synthetic,
Right.
to see if they're producing that kind of product.
Yeah, I totally agree that I think synthetic definitely shows you that that gene is important at some point in the life cycle of the organism. But that just because it's expressed doesn't mean it gets translated. And I think that that's one of the getting that complete translatome is one of the things we need to be able to predict.
I'll add on here that the mdr1 is regulating transcription, and so we are really interested in just what's being transcribed. So the transcriptome is the place to look for that. But yeah, it doesn't, just because something's transcribed doesn't mean it's translated. What other questions do people have?
Hey, Kaitlin.
Hi, Matt.
Nice talk. Thanks. Yeah, I was just curious. I mean, have you looked? I mean, you were kind of breaking down the different classes of TEs that were falling into these, differently imprinted and expressed genes. Did you look at gene functions too when you were looking at, the imprinted? I mean, what is there enrichment there, and what type of functions are we talking about?
Like the ontological terms.
Yeah.
They're enriched for not having any. So yeah, there's not a lot of information available there.
Yeah. Yeah, and I mean, is there information also available from these Arabidopsis studies that you were mentioning? And are you seeing kind of overlap of, similar loci being affected or across
Well,
species?
I haven't looked at the ontological terms for the Arabidopsis papers, but I do know that across, I think between maize and Arabidopsis, there's only a handful of genes. I know that between rice, maize, and Arabidopsis, there's only 11 genes that are conserved as imprinted. And I think there might be like a little bit more between maize and Arabidopsis, but either way, it's in the low double digits.
Okay.
So the conservation of imprinting is interesting, but for the functions of what has been conserved, it seems to be largely machinery that controls imprinting.
Mm-hmm.
So, yeah.
Cool. Thanks. Yeah, I really enjoyed the talk.
Thank you.
Looks like Maud has a question next.
Yes, I was wondering if you looked at core or dispensable genes, and do you know if your DE genes are dispensable genes, or is it known in Arabidopsis? What's the...
So in Arabidopsis, imprinting is largely thought to be a byproduct of Demeter targeting transposons. And I haven't looked at if these DE genes are core genes versus dispensable, but my expectation would be that they're probably dispensable. And that expectation also stems from, our previous work looking at the presence absence variability of imprinted genes and, the relationship with imprinted genes and mdr1, and that presence absence variation makes me think they'd likely be part of the dispensable genome.
Okay, thank you.
Thank you.
All right. Do we have any other questions out there for Kaitlin? Let's see any. All right, then I will call it. Thank you everybody for coming. We have 3 more Z evolutions before the end of the semester. And yeah, I look forward to seeing you all next week. Thanks again, Kaitlin.
Thank you.